immunostimulatory microrobots enhances anti-tumor immunity. In this project, we utilized the physical propulsion of micromotors coupled with an immunostimulatory payload for cancer immunotherapy. So immunotherapy has garnered significant traction in cancer treatment. The most successful of which is actually this immune checkpoint blockade, which are these antibodies that are used to block specific proteins on the tumor cells that the tumor leverages use to shut down our immune system. Through this mechanism, these antibodies essentially take off the brake from our immune system and unleash its potential onto these malignant tumors. While immunotherapy has been very promising, there are still kind of a lot of major challenges preventing their successful translation for every single patient. An example of these challenges include the fact that a tumor microenvironment is very immunosuppressive. So that even though the immune system is very active, once they get to the tumor site, the tumor cells are able to very rapidly deactivate these immune cells. In addition, the tumor is a very heterogeneous microenvironment, such that perhaps that you might not have the proper T cells and immune cells that can recognize every single different cancer cells. So trying to overcome some of these challenges, people have leveraged different types of approach in this combination immunotherapy fashion by combining different forms of treatment with immunotherapy in order to help enhance the efficacy against tumor. Examples of these therapies include radiation therapy, phototherapy, and also chemotherapy. Using these different forms of treatment therapy along with immunotherapy, which essentially targets different sides of the tumor cells, allows you to get significant increase in the efficacy and ultimately be able to cure cancer as a whole. In addition, micromolars are these tiny machines that can be designed to perform very complex tasks. And recently, there's actually been a lot of attention in the biomedical application of these micro machines and the clinical adoption. At the nanoscale, these machines can move extremely rapidly and are very powerful, that are made of different materials and versions that can have varying amounts of propulsion capability. So really, through this perspective, we wanted to see if we can leverage these powerful mobility as a new mode of action to disrupt and destroy solid tumors. And in order to achieve this, we designed an immunodestructive micromolar for cancer treatment. We hypothesized that the mechanical disruption of these micromolars can induce very irreversible damage to a solid tumor when they are properly injected into this very tight tumor environment. In addition to this, we also loaded the micromolars with an immunostimulatory payload, which are these outer membrane vesicles derived from a bacteria called E. coli in this case. They basically act as an immune stimulus and also in this combinational immunotherapy approach to treat cancer. Once these motor OMV are injected into a solid tumor, basically the micromolar propulsion will essentially lead to these tumor cell deaths and also damage in the tumor environment. And then the OMV that are released in this microenvironment will basically recruit and activate local immune cells into the site to not only process these dying tumor cells and be able to produce an immune response against them. Ultimately, this treatment at the primary tumor site is able to activate the immune cells in the host and basically be able to regress secondary tumors that are distant from the primary treatment site. The design of these micromolars are actually begin with these magnesium microparticles that are roughly about 20 micron in size. First, we actually started it with gold layer, and the gold layer is basically supposed to help with the micromolars to be able to propel much faster and act as a field catalyst. In addition, once the gold layer is deposited, we use atomic layer deposition to deposit the titanium dioxide layer. The titanium dioxide is just an inactive material, and basically what happens is that during this deposition process, we leave a very small opening that doesn't get this titanium dioxide due to the contact with the surface of this glass that we're using. And then subsequently, this opening basically can react with the environment and produces these giant micromolars that can propel in one direction. After the titanium dioxide, we coat it with a PLGA polymer layer and also this calazine layer in order to create this positive electrostatic charge, which basically helps this negatively charged OMVs to coat onto the surface, forming these immunodestructive micromolar in the end. So to confirm the proper fabrication, we visualize these micromolars through scanning electron microscopy and also energy dispersive X-ray spectroscopy. From the image you can see here, we can see a very clear small opening, which will actually direct the propulsion of these micromolars to that direction. In addition, in the elemental mapping, we saw that the magnesium and the gold is fully coated onto this micromolar surface. However, for the titanium dioxide layer and also the nitrogen from the calazine layer, we see like a small opening, which is basically the opening right here that demonstrates these unilateral directional propulsion of the micromolar. Next, we confirm the proper loading of the outer membrane layer onto these micromolars. And to do that, we actually use some fluorescent microscopy. We labeled the calazine layer with FITC dye, which is stained in green, and also we labeled the OMVs with this rhodamine dye, which stains them in a bright red. And basically from these brightfield images here, you can see that in the micromolar, it's overlaid with this green and also this red layer, indicating that these micromolars are fully coated with both the calazine and the outer membrane protein. Next, we went on to kind of optimize the amount of OMVs that we can load onto these micromolars by varying the amount of input in the fabrication process. So as quantified here, as we increase the amount of OMVs we put into the system for the synthesis, more and more OMVs get loaded onto these micromolars. At the 100 microgram per milligram loading, we were able to achieve the highest loading encapsulation efficiency, and thus we chose this ratio to move forward with all of our subsequent studies. Over here, you can see it's the immunoblot, a Western blot, that basically identified two major outer membrane proteins on the OMV surface, outer membrane protein A and also outer membrane protein C. While these outer membrane proteins cannot be detected on the bare molars, these outer membrane proteins are detected on both the OMV and also on the motor OMV final formulation, indicating that the protein profile is properly being transferred from the OMVs onto these micromolars. And lastly, we tested the propulsion capability of these micromolars. So the OMV coating does not really affect the ability for these micromolars to move in the different directions. And they were able to really move quite well in physiological buffers with a speed of about 30 microns per second. Since we are now both confident in the fabrication process and also the activity of these molars, we thought to kind of test the destruction capability. And to study this, we utilized these three-dimensional cell culture and grew tiny tumor spheroids that are about 1 to 2 millimeter in size. The cancer cells we employ here is actually fluorescently engineered to express this green fluorescence protein. So we can clearly see it in green here. So we make these tiny tumors with some different formulation, PBS at pH 5 in order to act as a propulsion field, and also on the active molar OMV here. We can see 4 hours after this incubation, the tiny tumor spheroid were able to be broken up into these tiny pieces and was significantly compromised upon this active molar treatment. Such morphological change was not detected in the PBS control group, nor in this static microparticle OMV control group. The static microparticle control is just prepared in a very similar fashion as the active molar, but instead we actually coated it with 2 layers of titanium dioxide in order to not leave any types of reactive opening. So here they are basically inert. Motivated by these encouraging results, we next tested these micromolars in animal models. And for this, we utilized the MC38 colorectal cancer cell line, and we implanted these tumors subcutaneously into mice, and basically allowed tumors to form over the course of 10 days. Then the mice with the tumor were treated with PBS, the static microparticle, and the active molar OMV on day 10. And then one day later, we dissected the tumors and also stained them for some histological sectioning. And basically, from the tissue sections you can see here, with the active molar treatment, the active molar is able to generate these tiny holes inside the actual tumor, where you can visualize the micromolars in these brown particulates here and also in the STAG microparticle group. The stark contrast is actually seen when we look at the entire tumor as a whole, where the active micromolar was able to create this giant hole in the middle of the tumor, something that we were not able to see with the STAG microparticle, and definitely not with the PBS control. In addition, to confirm that this destruction also led to apoptosis, we stained these tumor sections with some fluorescent tunnel staining to visualize the apoptotic cells. We saw that in the active micromolar group, we saw a bunch of apoptotic cells that we can visualize in green here. The nucleus of these cells is stained in blue. With promising mechanical destruction results, the next thing we did was we confirmed the error component of our formulation, mainly an immune priming. And to kind of give a little bit of background on how our immune system works, basically we have these antigen-presenting cells, such as dendritic cells, that are already scavenging for these foreign substances. The moment they take up these foreign pathogens and foreign antigens, these immature dendritic cells become activated and differentiate into these mature dendritic cells, which will increase the expression of these various surface proteins that can basically interact with different specialized immune cells in the adaptive immune system in order to train these immune cells to recognize these foreign pathogens. So to test the ability to activate the immune system, we utilize the dendritic cell line, DC2.4, and add various formulations to the cells. The mixture was kind of incubated for 2 days, and the supernatant was assayed for these frontline pro-inflammatory cytokines, TNF-alpha, IL-6, IL-12P4, and also IL-1 beta. Compared with PBS, the micromolars that are coated with the OMVs were able to have a significant amount of release in all 4 of these pro-inflammatory cytokines. Contrary to that, the bare micromolar, which does not have OMV coating, were not able to elicit any type of immune response, indicating that the immune activation mainly comes from the presence of these OMVs, as we can see here. In addition, we also monitor 4 different maturation markers, CD40, CD80, CD86, and MHC4. The upregulation in the protein expression of these 4 maturation markers is able to basically show that the immune cells have differentiated from the immature state to a mature state and be able to properly present antigens into the specialized T cells. Next, we tested these results in vivo in a tumor model. So similarly, we implanted a tumor on day 0 and let it grow for 10 days and basically administered them intratumorally with the different formulations. And then 1 day later, we dissected the tumor out for both to analyze the immune cells in the tumor and also to look at the cytokines that are being released inside the tumor. So looking at the dendritic cell population again, we were able to see similar upregulation of these 4 maturation markers, CD40, CD80, CD86, and MHC2. In terms of the frontline cytokines, TNF-alpha, IL-6, and also IL-1 beta, these are cytokines that are associated with innate activation. And all of these were upregulated upon treatment with both the active molar and also the STAG counterpart. Not surprisingly, we weren't able to see any differences between the STAG and the active micromolar because this innate activation mainly comes from the OMV and not so much the active proportion of the micromolars. However, we also analyzed the relevant immune cell population in the adaptive immunity. Because the adaptive immunity takes a little bit more time than the innate immunity to become activated, we studied these 4 days after the treatment rather than just a single day. For mice that are treated with the active micromolar, we were able to see a significant amount of CD4 positive and also CD8 positive T cells that are infiltrated inside the tumor. These infiltrated T cells are activated as quantified by the CD25 T cell activation marker, And a large majority of them actually expresses these interferon gamma positive functional phenotype, indicating that they can be actively out there and kill off the tumor cells. While we weren't able to see any differences in the frontline cytokines associated with innate activation between the STAG and the active micromolar, when we're looking at these IL-2, IL-12P4, and interferon gamma, which are cytokines associated with T cell activation, we were able to see a clear upward trend for the active micromolar that significantly outperformed the STAG counterpart. Since we confirmed that the molars can cause both irreversible destruction and also recruit immune cells to the tumor environment, we next studied the therapeutic potential in an actual tumor model. And to do this, we developed this bilateral tumor model where we implanted two tumors onto the mouse, one large primary tumor and also a second smaller distal tumor on the contralateral flank. 8 days after tumor implantation, we treated the mice at only the primary tumor site, with the different formulation every other day for a total of 4 treatments. And basically, as you can see here in the tumor kinetics, the treatment with the active micromolar was able to significantly control the primary and the distant tumor growth. And the really interesting result is that in the primary tumor, we were able to significantly eradicate 4 out of the 6 primary tumor, where the primary tumors were basically completely gone. For the STAG microparticle, we are seeing some efficacy, and this is mainly due to the presence of OMVs being able to overturn this tumor microenvironment and turning it towards more of immune activating. When we're looking at survival, the treatment of the active micromolars were able to significantly extend the median survival time to 38 days, compared to only 18 days and 24 days for the PBS and the STAG particle group, respectively. In addition, we also wanted to test the broad applicability of our platform in another colorectal cancer model. Because our technology leverages tumor antigens in situ at the tumor site for vaccination, the treatment should theoretically be able to control tumor growth in all types of solid tumors, as long as we can access the tumor and be able to perform intratumor injections. Here, we employed the CD26 colorectal cancer tumor and basically set up the tumor model in a similar setting. The results we saw was pretty similar, where the active micromolars were able to significantly reduce the tumor growth and completely eradicate 4 out of the 6 primary tumors, basically extend the median survival time and ultimately control the tumor growth, whereas the STAG microparticle was not able to achieve such profound efficacy. And lastly, we also tested the efficacy in a B16F10 melanoma model. B16F10 is a lowly immunogenic tumor, and it's very aggressive and grows very rapidly. As such, in this case, the STAG microparticle group is actually not able to have any sort of noticeable efficacy. However, using the active micromolars, we were able to see a better control of the tumor growth for both the primary and the distant tumor, where 2 out of the 6 primary tumor was able to be completely regressed. Throughout the treatment process for all 3 of these cancer models, we did not see any significant changes in the value weights, indicating that there was unlikely any major signs of toxicity associated with this micromolar treatment. To understand how the treatment of the primary tumor can lead to the regression of the secondary tumor, we derived sphenocytes from the mice that are treated with a different formulation, and we re-stimulated these sphenocytes with peptides that are encoding for tumor-specific antigens in order to be able to expand and proliferate the signal and perform analysis. 4 days and also 7 days later, we analyzed IL-2 and also interferon gamma, which are T-cell associated cytokines. After 7 days, we also took account on the number of T cells and was able to basically found that there was a significantly higher number of T cells that are specific for the ADPGK and also the P15E tumor neoantigen, in addition to the whole cell lysis, mainly for the active microparticle group. When analyzing the 2 different cytokines, IL-2 and also interferon gamma, we were able to see a very similar trend where the active microparticle was able to induce a significantly higher release of these cytokines to activate the T cells. Overall, these results demonstrate that treatment with lower OMV can evoke tumor-specific immunity systemically, allowing these systemically active immune cells to travel to the secondary tumor and be able to fight off disease and control tumor growth at that site. And lastly, we studied the safety of the more OMV platform and did not find any abnormal changes in the hematological cell counts and also common biochemical markers 1 day after treatment. Furthermore, one of the advantages of the coupling these OMVs onto micromorphs rather than using a co-administration strategy is that in addition to a synergistic effect, a free OMV can very rapidly leak into the circulation and thus increases the release of these pro-inflammatory cytokines in circulation. The release of these pro-inflammatory cytokines is undesirable as it can actually cause things like sepsis and also cytokine storm syndrome, which are related with the toxicity of these bacterial OMVs. So in summary, the movement of micromorphs can disrupt solid tumors. And by adding an immunostimulatory payload into this destruction site, we can recruit immune cells to process the dead tumor cells and activate the immune system. This combination approach can not only significantly control and eradicate tumor, but also evoke a systemic immune response that can not only fight off tumor at a distal site, but potentially be able to prevent metastasis. Ultimately, there are various different amounts of micromorphs, and hopefully we can leverage these as an additional tool in the combination immunotherapy treatment for the various solid cancer. So as acknowledgement, I wanted to thank my lab, basically Professor Liangfang Zhang, and also Dr. Yifeng, which are both integral to this entire project. And in addition, we couldn't be able to do this project without the Professor Joseph Wong's lab and all the experts in the field, as they are the micromorphs experts here. And lastly, I do have many financial funding and support that was able to really help make this project possible.